proceed to Professor Pieter Cullis from British University of British Columbia in Vancouver, who will present us new data about in two fields. One is the Marqibo as a lipid nanoparticle formulation of Vincristine for acute lymphoblastic leukemia, and a lipid nanoparticle formulation of siRNA for silencing genes in the liver. Preclinical and clinical results.
Okay, first of all, I'd like to thank Pete for the invitation to come here. I'm presenting on two topics, so he gave me twice, I think 20 minutes instead of the 10, and I'll try and confine my remarks to that interval. So today I'm going to talk about two products that have made it through to being approved by the US FDA. One of them based on small molecules, one of the small molecule formulation of Vincristine, a liposomal formulation of Vincristine, and the other an siRNA formulation that's against the liver target. As you're all aware, I think I have a pointer here somewhere. If that works. The problem that we're facing here is of considerable intricacy. We're trying to get an injectable of nanoparticles that will go around, circulate around in the body, reach some target site, get out of the vasculature at that point, and then in the case of intracellular delivered products such as siRNA, get to the interior of the cell. So this is not a straightforward proposition. Viruses have been trying to do this for almost 1 billion years, and we're trying to solve it in a time that's considerably less than that. Now, just a few mentions of the advantages of lipid nanoparticles. You've heard quite a bit of that in the last while. But the basic points remain, and I'm just going to repeat them because they're profound. They're self-assembling and biocompatible systems. They're really direct analogs of systems such as lipoproteins and vesicles that are used to transport in nature. There's really straightforward ways of making these systems. It's highly flexible. We can control size, circulation longevity, drug retention, fusiogenicity. And most importantly at this stage, they're by far the most clinically advanced of any lipid nanoparticle system with now eight approved drugs that have been approved by regulatory authorities. So these are the eight approved drugs. The ones in yellow are ones that we've had something to do with. The most recent formulation is the one I'll talk about, a lipid nanoparticle formulation of Vincristine called Marqibo. That's been approved for acute lymphocytic leukemia, and was approved in the U.S. in August of last year. The bulk of these systems here are for small molecules and primarily for cancer. There's also a vast variety a large variety of systems that are liposomal systems, lipid-based systems that are in clinical development. This just indicates a few of them that, again, we're directly involved in. So formulations of small molecules on the top part here, the top four, and then the bottom four are formulations of siRNA, all of which are in the clinic at the moment. And I'll focus my remarks today on the lipid nanoparticle formulation of transthyretin, which is just in phase 2 trials right now, with actually the results are going to be reported soon. And so why is this technology useful? In such a general way, and the points here are that we can enhance the potency and reduce toxicity of small molecules by increasing the amounts that's delivered to tumor sites, and these for treatment of infection, inflammation, and tumors. And also we can reduce delivery to healthy tissue and reduce some of the side effects. In the case of genetic drugs such as siRNA, we can protect the siRNA from degradation. We can get more of it to where we want it to go, and we can facilitate intracellular delivery. And we can also take advantage, the third point here, and I'll be making particular note of this, advantage of what we might term natural or endogenous targeting mechanisms to access cells that we might want to access. So how do you make these systems? I'm going back to basics here, but the point is that we're dealing now with a fairly mature technology, and we have fairly mature ways, fairly well-established ways of making material both at the bench scale and at the scale-up levels. Extrusion is a top-down approach where we take larger systems and make them smaller. We can work down to about 60 nanometers in diameter quite well with these lipid or liposomal nanoparticles. If we want to we'll use a bottom-up approach. We can use microfluidic mixing. This is a recently introduced procedure that involves just mixing lipid dissolved in ethanol or another water-miscible organic solvent with water, and then putting it through this. This is a staggered herringbone micro mixer. Out the other end, you'll get essentially limit-sized systems that are the smallest size that are compatible with the molecular makeup of the material. We can get down to about 20 nanometers or so diameter with these systems. How do you load systems? A pH gradient approach has now been well established for around about 25 to 30 years, and this is an extremely general and very potent technique whereby we can accumulate and maintain, say, up to 100,000 molecules, small molecules, or anything less than that within a 100 nanometer system. So again, a very well established way of doing things. How can we encapsulate larger molecules so we can deal with small molecules such as anti-cancer drugs? How about something with a molecular weight of 13,000 such as siRNA? And again, this is now a fairly routine process. We can use systems such as the micromixer. And here we're mixing the material up, the siRNA, with a mixture that contains a positively charged cationic lipid, and then run that through this apparatus. The mechanism behind this is we're ramping up the polarity very quickly in milliseconds, mixing tens of nanoliter quantities. There's an immediate electrostatic association between cationic lipid and the anionic polymer, forming these hydrophobic systems that precipitate out of solution first, and they're subsequently coated with more soluble elements as they fall out of solution to form the lipid nanoparticle system. So you can see how it is. We can get very high encapsulation levels. One can do molecular modeling on these. This is work done with Pieter at the University of Calgary to establish you have a kind of a current pudding sort of arrangement, inverted micelles inside this system containing the siRNA. So we need cationic lipids in order to load siRNA into lipid nanoparticles. That's a problem because these things tend to be toxic if there's positively charged systems in the circulation at physiological pH values. We've developed ionizable cationic lipids that allow us to they're only charged at lower pH values, not charged at physiological pH values. So it allows us to perform the encapsulation at a lower pH and then subsequently bring up the pH to physiological pH where one has essentially a net neutral exterior. And so the point that I'd like to emphasize here is that we now have siRNA systems generated by these techniques that really satisfy the basic criteria for in vivo use. There's really no point taking something in to even preclinical studies unless it satisfies these basic criteria: near neutral, relatively non-toxic, variable diameter, high encapsulation efficiencies, monodisperse, a variable number of oligos, scalable and reproducible. So just now let's talk about the applications. There's really, the applications we're talking about here are for systems that have no targeting information on the lipid nanoparticle exterior. We take advantage of the endogenous targeting processes that I mentioned, both for the small molecule system where we have long circulation lifetimes which lead to accumulation at sites where the vascular is more permeable, as well as the lipid nanoparticle, as well as the siRNA systems, and I'll indicate what goes on there. So for small molecules, the situation is relatively simple. All we have to do is get the material to the vicinity of, say, a tumor, and then let the material come out, that the small molecule come out to affect the disease. The system, the drug that we can focus on here is Vincristine. It's one of the most widely used anti-cancer drugs, 2 million doses a year, almost always used in combination with other drugs. So this is Marqibo. This is the nanosystem. It's about 100 nanometers in diameter. It contains 10,000 vincristine molecules, and it has a therapeutically optimized T-half or drug release of 24 hours. And just to point out the efficacy of these systems, the system here is actually telling me I'm supposed to end, but I think I have another 10 minutes. It's there's something blinking. Okay, great. I was going to say I really only got started here, but just to point out the efficacy of these systems in a animal tumor model. This is a tumor model injecting 1 million cells into the flank of the animal, wait a couple of weeks until the tumor size is 100 milligrams or so, and then inject via the tail vein. We get much more to the tumor site as a result of long size than we do with the same injection of the same amount of the free drug, and we get dramatically improved efficacy as a result of that. This also applies to people. There's been a number of studies done now. I'll just indicate one of these, which is a phase 2 study in front lines where essentially vincristine was substituted, or the LNP vincristine was substituted for the oncovin, the O, which is the drug in CHOP, cyclophosphamide, doxorubicin, oncovin, and prednisone. So in this clinical trial, just to point out the results that came out in 2002, this has been an ongoing process now for many years. This is one of the problems with developing a drug. It takes forever. But the overall survival at 2 years, 70% with this historical controls and 98% when we included the oncovin, the Marqibo within that formulation. So this addition can have profound effects. Now, since then, a company named Talon Therapeutics has been pushing this drug through for approval in acute lymphoblastic leukemia at third line. And so this is just indicating that this is the part that's bracketed here. Third line this is as a single agent. Single agent therapies historically induce very few responses. The 2-month median objective survival, highly toxic. A lot of people that die right at the beginning of the therapy. And just to indicate what was observed with Marqibo in the same cohort of patients, it was a overall 35% response rate with a 20% complete response rate. On the basis of this data, the FDA approved Marqibo for ALL, acute lymphoblastic leukemia at second or greater relapse, and there is now a registration trial that's been initiated for frontline non-Hodgkin's lymphoma, and I suspect that's going to be, well, at least that has been initiated last year. This is there's a very large dose intensification and also toxicity reduction as a result of the nanoparticulate delivery system. Now to go on to siRNA and the lipid nanoparticle systems to enable a genetic drug, large molecules such as siRNA, are more complicated. And obviously in this case, we can't just get it to the vicinity of a target site. We have to get into a target cell and then inside that cell. And so this is a more complicated situation. We use here a very convenient animal model, which is the factor VII model. So we can inject our lipid nanoparticle containing factor VII against siRNA by the tail vein into this model or into a mouse, and then assay for factor VII in the blood. And so factor VII, as you're aware, clotting factor, it's made in hepatocytes. And so by 24 hours later, we can examine the the plasma, the 4 factor 7 levels, and get a pretty rapid readout as to what sort of knockdown we're getting, what sort of silencing we're getting in this for the particular formulation we're using. So we're using a so-called non-targeted system, lipid composition with cationic lipid about 50%, and then other structural lipids, DSVC, cholesterol, and a PEG lipid in those ratios. So what we found in using this this relatively high throughput model was that the gene silencing potency of these siRNA systems was very sensitive to the species of the cationic lipid. You'll see here that the head groups look pretty much the same, but the activity is really quite remarkably different. We're looking here at the dose levels that are required to induce 50% gene silencing or factor 7 gene silencing, and we see in one case for the DMA with the ester linkages, we're up at 10 mg per kg, 1 mg per kg for the same compound with ether linkages, and then we move to this KDMA lipid, we're getting down another factor of 3 or 4. So this was something that we took seriously, or took seriously as one way, with something was a bit of a crack in the door that we exploited. And so we've synthesized and screened over 300 ionizable cationic lipids to see which ones are the most potent, and we identified really a quite remarkable pH sensitivity, pKa sensitivity here, where the most active lipids have a pKa of 6.4. And so so this is, you can see you can move almost 3 orders of magnitude by changing the pKa from 6.4 down to 5 or lower, or 8 or 7 or higher. So the understanding that we have of this is that these cationic lipids combine with anionic lipids inside the endosome as the pH is reduced, causing formation of non-lamellar structure and release of the siRNA. The bottom line here is that we've moved from systems where we're over the last 4 years, where we have potencies in the range of 1 mg per kg, down to systems where as 5 micrograms of siRNA per kilogram body weight to silence genes in hepatocytes, and by extension to silence any other gene in hepatocytes they might want to silence. So this is currently the gold standard lipid. That's this MC3 is the gold standard lipid that has this capability of maximum bilaterally destabilizing capability and optimized pKa value with these very low or very potent gene silencing capabilities. Now you may be asking, this is all very interesting, but what's the targeting mechanism? Why are they going to hepatocytes? And this is the virtue of doing experiments in vivo. You sometimes get very lucky if you can make biology work for you. And I'm not going to go through the data, but we're using PEG coatings here that come off very rapidly, half times for the PEG lipid C14 anchor from dissociating on the order of minutes. There is an association of ApoE subsequently with these particles, and then these particles are then they're treated essentially like chylomicrons as fats get taken into the body. They're taken up into hepatocytes via the LDL receptor, scavenger receptor, etc. There's a number of receptors that will accumulate these into hepatocytes. There's substantial data to support this. I'm not going into it. What I'm going to do is just indicate the properties of these systems. For one, there's 3 indications now. 2 of these are using this MC3 lipid. This is a systems for hypercholesterolemia silencing PCSK9. This is work being done in combination with Alnylam Pharmaceuticals. And the other one against transthyretin-mediated amyloidosis, which is in phase 2 clinical trials at the moment. Now, just a word or two on transthyretin-induced amyloidosis. Transthyretin is a vitamin A transport component in the blood. It forms a tetramer. If there's any one of almost 100 identified mutations, it can cause fibrils to form. These can be deposited in nerves and heart and other regions to have very deleterious effects, to say the least. The only cure presently is a liver transplant, and it's certainly a very large unmet medical need. The study design here is on healthy volunteers, a phase 1 study. There's a single-dose escalation study, 4 subjects per cohort, and a single 60-minute IV infusion of the lipid nanoparticle containing siRNA against the transthyretin. Obviously, primary objective, evaluate safety, but secondary to look at the pharmacokinetics and pharmacodynamics, serum TTR, et cetera, all of those. That was completed last summer, and with very positive results. This is just indicating here the dose dependence, 0.05 mg siRNA per kilogram body weight. There's not too much effect on the TTR, the transthyretin protein levels in the circulation. However, at 0.15, one can see a dramatic reduction, and higher, one can see a dramatic reduction in the levels in the blood, which is maintained over 40 to 50 days. So you can see the possibility of single injections or bi-monthly injections to keep siRNA, the TTR levels in the blood at very low levels. So in summary, this, as it's called by Alnylam, ALN, TTR02, safe and well tolerated, no significant adverse events. Rapid, dose-dependent, and durable and specific serum TTR knockdown in these following a single dose. There's one other point which I didn't put the slide in to save time, but there's an excellent correlation of TTR knockdown in patients with the TTR knockdown at the same dose in non-human primates. So you're almost getting a clinical validation in a preclinical model, which is really quite remarkable. The phase 2 data on this is expected at the end of this week, and we're hopeful that the results are going to be positive. So just a word or two about the future. For small molecule drugs, it's now pretty well validated that for all anti-cancer drugs, in particular toxic drugs, there is real reason to move to lipid nanoparticle-based systems for reasons of reduced toxicity and enhanced potency. There's obviously some substantial commercial opportunities here, which we in the field have been aware of for quite some time. And in the case of genetic drugs such as siRNA and increasingly messenger RNA, there's enormous potential. We can essentially silence any gene we want at the moment in hepatocytes in people with very reasonable therapeutic indices, and the range of potential targets is certainly not limited to this. There's now increasingly systems where we can hit and silence genes in a variety of other tissues, immune cells, distal tumors, bone marrow, endothelial cells, brain, etc. So one can expect this third modality for treating disease, in addition to biologics and small molecules, to have a very vibrant future. So with that, I thank you very much. I'd like to just acknowledge the work of a very large number of people, both at Alcana, who did much of the synthesis of the cationic lipids, Precision Nanosystems, who were working on the microfluidics approaches, at Techmira, Alnylam, a lot of chemistry done there as well, UBC chemistry, and physics, microfluidics with Carl Hansen, and my own lab, who work more on targeting other tissues than the liver. So with that, thank you very much.
Congratulations to your work, which has really shaped the field in the past years. Yes, please.
I have 2 questions. First question, Did you already use the microfluidic method to manufacture the products for the clinical trials? And the second question is that I noticed that you used the sphingolipid in the formulation for Marqibo. So what's the advantages of sphingolipid compared to the traditional liposomes like Doxil?
Right.
Thank you.
Yeah, the answer to the first question is no. We've been used, the technique that's been used there is a T-tube. It's a related process, but we haven't used the microfluidics approach for the clinical trial material. With regard to sphingomyelin, the reason that was included was in order to get the right payout rate for Vincristine. We wanted to get a half-time for release of about 24 hours. That was optimal in our animal models, and the formulations containing other saturated lipids didn't give us that payout rate. So that's the reason it was chosen.
The endogenous targeting mechanism that you alluded to is very interesting. The LDLR has also, depending on the details of that mechanism, been associated with BBB targeting too. It should be a significant off-target effect. That's the ApoE.
Well, we were hoping for that off-target effect, but we didn't see it.
Were you hoping as a target effect or an off-target effect?
Well, we're hoping to see some delivery across the blood-brain barrier, but we've never seen that.
You haven't seen any delivery onto the barrier? BBB?
No, we haven't looked for that, but
that would be something to think about, I think.
You said that the PEG lipid partitioned over here at the site. The PEG lipid partitioned off of the liposome quite rapidly and then was replaced. So why are PEG lipids widely used on liposomal formulations? Yeah,
we're not using the PEG lipid here to give us long circulation lifetimes. We're using it to get the right size of the systems as we make them using these bottom-up approaches, such as the microfluidics approach. If we didn't have the PEG lipid there, we just see large aggregates. So, I mean, the PEG lipid is on the outside. It's deciding, A, the size of the system, and B, the stability. So if we didn't have the PEG lipid there, we would see a lot of we'd see massive aggregation in these systems. Right, but subsequent to that, you don't really want the PEG lipid there because it inhibits uptake of these systems into target cells.
But then in other liposomal formulations with PEG, does that PEG also partition off immediately, and why is it added?
Oh, no, in the other, so we're using a PEG lipid that has a short acyl chain, which means that it can come off quickly. And the PEG lipid that's used in drugs such as Doxil has C18 chains, and that comes off and then doesn't really come off days or longer.
Okay.
Yeah, Pieter, the real beauty of this Vincristine formulation with the cholesterol, sphingomyelin, was this payout rate. And the drug is now released into the bloodstream, which then is like a bystander leakage for the ALL, and you don't have to have extravasation.
Yeah.
But could you increase you've only got a 20% complete response rate. Could you increase that again by dosimetry, by targeting these liposomes to the actual ALLs?
Yes, yeah, and these are the kinds of targeting approaches. I think we're using the blood compartment that's the easiest one that could go after, and a lot of the issues that we face with targeting could maybe be avoided in that particular instance, but we haven't done that yet.
Thank you very much for this interesting data. I'm also very happy because Sanofi is co-developing this Alnylam TTR02. Right. Just one question concerning the phase 2 recruitment. Because you said the PEG, it's not, the objective was not really a long circulating nanoparticle. So that means, having said that, one can imagine a macrophage uptake, and then there is a possibility of macrophage saturation at some dose. And this may pop up severely when we go for repeated administration in phase 2. So did you take additional measures in fixing or making this repeatable dose regimen by taking into consideration the probability of macrophage saturation and then macrophage recovery before you inject the second dose so that the patient can recover from any immune issues?
Yeah, we haven't seen any macrophage inhibition as a result of uptake. I should mention these are co-dosed with or pre-dosed with dexamethasone before administration, so there hasn't been reported in the patient population whether that's due to the dex or whether it's just that these things are. I mean, these things are certainly relatively immune silent in the preclinical models. That's clear. Yeah, we wouldn't suspect there's going to be any problem with any compromisation of the immune system due to uptake by macrophages. These things go remarkably avidly to the liver and to hepatocytes. So.
Okay, thank you very much.